Daniel Leskovar is a good friend of mine. He's also a professor at Texas A&M University, and he works out of the Uvalde location and has been working on vegetables. And he's going to talk us through some of the things that focus on watermelon performance. So with that, Daniel, if you'll take the time, happy to have you here.
Perfect. Yeah. Thank you for the opportunity to, and good morning, everyone. My name is Daniel Leskovar. I'm a professor in vegetable stress physiology at the Uvalde Research and Extension Center. So when Dan asked me about this presentation, so what I thought what we'll do is first give you a very quick overview, some general issues and challenges that we are facing in this region, which you're going to see that are not much different from what you are facing in relation with the droughts and climate changes. And then I'm going to go and talk a little bit about the important aspect of watermelon systems, one related to the seed quality and transplant quality, and then address maybe 4 cases in relation with crop management, particularly water management in relation with other 4 practices. Most of this work has been done through different graduate students in my program, and I will try to emphasize some key points on those. Here is a map of Texas. What you see the arrow is where our location is in the Uvalde. It's in a region called the Winter Garden because of the mild climate. We can grow a lot of leafy greens as well during the winter, spring. Here is where the main campus is, Texas A&M University. And then these 3 regions are the main production region for the horticultural crops, citrus, sugarcane in the Rio Grande Valley. Ours is a combination of vegetable crops and field crops. And then the high plains is a very, very large area that is mostly monoculture in terms of wheat and cotton and corn. And here you can see in the west, the very, very nice Santa Elena Canyon that separates, this is Texas wall, this is Mexican wall. So it's, this is as close as we can get as Utah. But it's a very nice area. So our region, the Winter Garden, it really is very, very diverse and dynamic. We have really good, excellent soils and water quality, the mild climate. We have a balanced crop rotation systems. The economy in general is pretty stable, and we have these dynamic corridors that goes all the way from the border with Mexico all the way to San Antonio, Austin, Laredo. One of the key issues I want to stress is the water resources. So our region basically depends on underground water aquifer called Edwards Aquifer. That aquifer serves the city of San Antonio and the Winter Garden region. The city of San Antonio is the largest, the highest growing city in the United States. It's about 1.5 million, and it's incredible the growth of that. So as such, this aquifer provides the municipal water for the city and also for the ag sector for us. So there's a constant dynamics between the users of the water, and in fact, the Edwards Aquifer is one of the most highly regulated aquifer in the nation. The growers are metered; they can only pump 24 inches per acre. They can use more, but reduce the allocation to another acre. But water is actually our major commodity. You can buy, transfer, sell, store it. Bank water, and so there's a lot of market associated with the water. So Dan talked a lot about climate change. We are facing the same situation. Temperatures increases, average increases, and tremendous drought. This is just, I put it the one in July, but you can see here July in the middle of the summer, Texas is an exceptional drought. Right now, the city of San Antonio, which received 30 inches of water, they have 1 inch in the last 6 months. So this is a very, very critical situation. With that, the region has been forced to improve irrigation technologies. Back in 30 years ago, mostly all irrigated crops were by furrow irrigation or flood irrigation. And then 20 years has been changed a lot, particularly using more center pivot or linear systems, and much less in terms of the drip irrigation. But this was a big push in relation with the dynamics of the water in the region. This is an example. It was 30 years ago how watermelons were grown by the growers. Direct seeding, mostly the seeded watermelons or diploid or 2 eggs using furrow irrigation or very low efficiency, very non-uniform. And then about 20 years ago, the change came very rapidly. This is an example of a very successful grower that uses containerized transplants, subsurface drip irrigation, and this was in a mid-texture soil about 30 miles from our region. So in addition to the irrigation technology that I mentioned, like drip, there are other practices or strategies that can be used, as Dan also mentioned in presentation. So how you manage stress, for example, using plasticulture, using soil amendments, using improved irrigation based on crop coefficients. What happens if you put less water and how that affects? And then from the plant size, you have two aspects to it. One is looking at how if you grow watermelon in the different cropping systems, using tillage difference, using cover crops, using windbreaks. And then from the genetic point of view, or the variety point of view, the breeders are constantly looking at ways to improve the drought, the quality of the plant growth and food growth and food quality, but also looking more lately in terms of how plants adapt to heat stress, in terms of how the leaves adapt. And one area which is important is in terms of the root systems. Dan mentioned basically how you, very nice presentation in terms of irrigation requirement. So basically the formula was mentioned, but here we have a classical for watermelon, we had to come up with a crop coefficient that you can multiply that with a reference evapotranspiration. And then you have to consider somewhere how effective is the rainfall. Is it 50% effective if you have mulch? Is 20% if you have a seeding or transplants that are not really developing food systems? So that's an aspect that you can consider for the irrigation requirement. And if you don't have a weather station, I imagine Utah is very well positioned for looking at the nearby weather station for taking the evapotranspiration or ETO or the reference. If you don't have, you can get this simple device called anemometer, which basically provides a very good representation of the crop evapo reference one, and then you can use that data and then multiply by the crop coefficient, like Dan nicely explained. So if you don't use like the coverage of the plants, one possibility is to start with, be using there is a very good publication, it's free, it's FAO 56, that for watermelons, they have this 3 crop coefficient, low in the beginning, at the peak, and then going down close to maturity and harvesting. Now, an important aspect in terms of the productivity quality is that the response of plants really are based on the interaction between genetics or varieties, the environment, and how you manage the crop. So let's say environment, when do you grow the crop, whether it's in the spring or in the fall? Where do you grow the crop, whether it's in a sandy soil or silty clay soil? How do you grow the crop, whether you use different spacing, you use a plastic mold, you use different irrigation systems, together with the varieties or genetics, that will provide you really the answer for the expression of those plants in terms of productivity, but also in terms of quality. And we've been working a lot in terms of phytonutrients in fruits and vegetables, particularly also in watermelons in many years back. This is an example of the genetics. This was my first PhD student, Heijin Bang, who basically looked at the genetics of pigmentation in watermelons and trying to elucidate what we call the carotenoids pathway in different melons. It's a fascinating work. But so that's one aspect. You can certainly genetics is a big part. But also the phytonutrients of quality can be affected by other practices like deficit irrigation. And we've done some work with her in terms of what happens if you use deficit irrigation, 75% evapotranspiration or 50%. So in general, you see that in not only watermelon, but other crops, you can see that there is a trend towards increasing the phytonutrients, but there is a penalty, and the penalty is a reduction in the productivity. How much reduction will depend on the level of stress imposed on the plants. So with that, let me just go and talk a little bit about now as a aspect related to watermelon seed and transplant quality. And I will tell you why it's very important. You probably know already. Number one, it's very expensive seed, right? Especially the seedless watermelons. But then also in providing 4 cases studies that we have, in terms of the irrigation management in relation with these practices. So if you grow watermelons, I'm sure you've seen in the nurseries, or if you grow on your own, when you use a triploid 3X or seedless watermelon, it's very common to identify those varieties because the cotyledons, the seed coats are going to be attached to the cotyledons, most of them. And why is that? Part is because of this cross, how these 3X or 3 seedless watermelon were produced. Basically, it's a cross between 2 and 4. You get this plant which get seeds, and these seeds produce fruits without seeds. And the reason why this happens, and the reason why the vigor is also very small, is by doing this tetraploid and diploid, basically is what we call a chromosomal imbalance. And by that chromosomal imbalance, it changes the composition of the seeds, both physical composition as well as chemical composition, that affects the reserves of the embryo that they have to emerge and produce a very healthy seedling. So the energy of these seeds coming from, in the nursery, you will see that they don't have enough just to really release the seed coat. In the diploids or seeded watermelon, you will not see. And many years back, this was a huge problem and continues to be to less extent because the seed technology have improved, but it was not that uncommon like 20 years ago, and you still see sometimes issues pertaining to abnormality in the triploid watermelon. So this is the very extreme cases, very, very, very small roots, elongation, sometimes you have 3 cotyledons and so forth. And part of that, we have a graduate student that work on the seed anatomy, physiology of the triploid watermelons, and a couple of points that I wanted to mention for you to know. The difference between a diploid or seeded watermelon versus a triploid, you see this layer here called ED, endodermis. Here is the embryo, and this is the hypodermis and epidermis. So what we found is that this layer is actually it's a multi-cell layer, very thick layer, that disrupts the water uptake coming upon imbibition of the seeds. Number 2 is you see this cavity called seed cavity. In many of the seeds, particularly medium vigor or low vigor watermelon, you see the seed cavity is almost empty. So between the physical conditions of the endodermis and the cavity, basically this seed, for example, did not germinate, as opposed to these seeds that you have the embryo pushing out towards these tissues. Another, don't worry about the numbers and what the tables in general, but the message here, these are different gas exchange in seeds. This is from diploids, and these are the triploid, different vigor, low vigor, high vigor, and 3X, and low, medium, and high. There is a gas, obviously, seeds upon inhibition exchange CO2 and oxygen. And then the ratio of this is called the RQ. When you have an RQ of 1, close to 1, that means there is a really good equilibrium between the CO2 release and then the uptaking. And this is mostly relates to resources on the seeds associated more with sugars, we call polysaccharides. But in this case, on the triploid, particularly the low vigor, has a lower RQ. This lower RQ is an indication that possibly that the energy of these seeds is not based on carbohydrates, but is based more on lipids, fatty acids, and potentially proteins as well. So going back to transplants, so why we may have problems with transplant quality? The bottom line is that transplants are grown in containers, and then the root confinement is a big factor affecting the morphology, the physiology of the plants. These are high quality transplant. This is a watermelon, and this is a low quality watermelon transplant. You can see the seedlings are elongated. They already begin to show male flowers. So this is an indication that these plants are getting either old or surpass the time for shipping to the grower. With that, so one of the aspects is if you have a very weak transplant under high temperature conditions, basically you have transplant shock, right? So that the plants do not compensate the transpiration with water uptake, high temperatures. But if you have a healthy seedling, this is what you expect. So we've done a lot of work using abscisic acid, which is a natural hormone in plants that regulates the opening of these small holes in leaves called stomata. By doing some application, we were able to control the height of the transplant, so produce a compact plant with a very good balance between shoots and roots. This is an example of one variety. This is another variety, and you can see basically the height separation was anywhere from 10 to almost close to 20%. And you can do that by applying this either a few days before delivering or even up to 3 weeks before delivery. The bottom line is make sure you always start with a good and high quality transplant, as you can see here in the picture. Now, we've done, so how we can improve the transplant quality? We've done quite a bit of work on a very unique humic substance, which is a solid material that we've been working for almost 5 years. Applied to the media, and you can see here these humic substances, these trays of watermelons that have the humic substance versus the control, and then you can see here the growth in terms of the leaf areas compared to that. So it's a very significant response. We've done in several species, not only watermelon but also lettuce, tomatoes, and peppers. And then we were looking at both the root systems and shoot systems, and overall we see an enhancement of root systems in mostly of the fruit crops, but less, a little, initially less in lettuce, but later on, after transplanting, we can see a big boost. And when we look at all the productivity, basically we have a positive response, and watermelons was by far the higher response. So by manipulating the transplant quality, we were able to improve the productivity of the plant. And this is just an example. One of the varieties that we use, this HS is humic substance, and you can see that once the trays have the humic substance at the beginning, not much change between the control and humic substances. But once the transplant are going into the field, here the red line, you can see the boost in the root development. Here is root dry weight, but also we've seen other root traits compared to the control. And then interesting is that when we put these transplants under different heat or drought stress, you can see here is at 50% irrigation, simulating drought, is more productivity, and this is at 100% irrigation, and the cool conditions increase that. When you put the very, very hot conditions, obviously productivity goes low, but still we have a trend under heat stress and drought stress. Sorry, this is very heavy, but I want to point out this point here. This is the watermelon. These are the different shoot and root traits. And importantly, as you can see, when you see green arrows going up, that's a positive response on many of the root traits after transplanting. So this is post-transplanting measurements. This is for both for the seeded watermelon and for the seedless watermelons. So what we concluded, the root performance were outstanding in the fruit-based crops, peppers, tomatoes, and watermelons. And those perform better in terms of productivity, particularly under abiotic stresses. And in part, this has been related to mostly increases in root length and root surface area. And we know that root length and surface area are traits involved in nutrient and water uptake, that eventually this is translated in better productivity. So conclusion is that humic acids, the one model humic acid that we use, I mean, there are many in the markets and many really don't have these responses. But some of these applying into the media, they have some possibility for enhanced the transplant quality. Now, moving into the quickly on irrigation, we've done in the past several deficit irrigation studies in watermelon, looking at different rates, using different varieties. We have 2 diploids, 2 triploids. And here's an example of what happened with deficit irrigation. So this is a well water treatment, this is 75. So you see difference in the canopy associated with that, which is no surprise. But importantly, okay, how that affect the fruit development? You can see here that in the diploids, the 2X watermelon, you can see a larger reduction in size as compared to the control. because of the triploids, the watermelon normally are much more oblong to round. You don't see that change that much. You see a reduction, but it's not as prominent onto the diploid. So this could be actually be used as a potential tool if you have a very large triploid watermelons and you want to produce a relatively smaller watermelons, approaching a kind of mini personal watermelons. It's not difficult to use some of these techniques. And when we look at the productivity of the plants, we've done this in several locations in Texas. You can see that the penalty is not that much. If you reduce 25% water applications compared to 100%, you really don't have a significant reduction in terms of productivity loss. Now, what about the color of the fruit, particularly lycopene, which is a carotenoid that has been used as an attractive trait for consuming watermelons? Interesting, irrigation rates really don't have much effect in terms of changing the lycopene content. The more importantly is that you need to harvest the watermelons at the ripe to semi-ripe stage if you want to maximize lycopene, which is associated with color. And this is regardless whether you use deficit irrigation or you use well water conditions. Another aspect that is very important, it was covered by Josh, and is in terms of cover crops and tillage systems, and Dan also talk about that. So strip tillage is a practice that we've done some work for 3 years. The bottom line that we compare using strip tillage versus conventional tillage is that we have an increase of productivity, particularly the high value watermelons in the terms of 11, 17 pounds. This is very consistent on the 3 years of the study. And importantly also is that consistently we have seen about a 5.5 point increase in the sugar content of the fruits. And this is actually significant because, as you know, melons or watermelons, the sugar content is a critical factor for the retailers and for consumers to really consume this wonderful fruit. Now, we've done a lot of work on biostimulants and a lot of work that we really didn't see any positive responses. A lot in the market, they're huge. Now we've done studies for Trichoderma and Bacillus, either deep in transplant, drenching, injection, and at the end of the day, we look at the field, and then before even we continue harvesting, we wonder, and bottom line is that it's very difficult to see differences with all of these studies, mostly because we have not imposed stress. Once you have stress, you may possibly begin to see some benefit from these biostimulants. This is an example of a kind of biostimulant, but it's based on nutrients and organic matter. And we've done some work under different stress scenario. We're using drought stress with deficit irrigation and applying in different timings and drenching and application through drip. This has been the vines, and then here we see a very interesting that this biostimulant actually increased productivity. It's called vigorizer of 6 candles compared to the control and the deficit, and also and the 100% irrigation. So there are some materials that may be more than one particular biostimulant type, whether it's biological or chemical, but when you incorporate nutrients can really have a good response. And finally, I wanted to talk about humic substances, like I mentioned about the transplant, but as you know, humic substances have important effects on soils and plants. There's a lot of literature on that. For example, in the soils, increase soil water holding capacity, the permeability. Critically important is increasing soil organic content. And this also has a relationship with also improving soil health in terms of microbial community. At the plant level, we are interested to see how these organic sources of humic substances also improve root development, for example, and also how they protect the plants against stresses. And this is basically reducing the oxidative stresses that the plants have, producing what we call reactive oxygen species. I have a student in the master and PhD on this project, and did a lot of work on studying the incorporation of this solid material in the beds. He's establishing here a crop, I believe, was peppers and then later on watermelons. This was watermelons. So basically, this is the solid material. It's lignite based. So you incorporate in the planting bed and then do the mulching and then ready for transplanting. This was experimental fill out, small field that we use deficit irrigation as compared to well water, and then evaluate the response of diploid and triploid. And the bottom line is that overall in this 2-year experiment, we've seen that there was a positive response anywhere from 15 to 20% in 1 year and about 5% in the second year, which this year was very stressful, but we still were able to see some increases. But you can see that either at the 50% irrigation or deficit irrigation or well water, we see really a good productivity response. But also, and importantly, is we can see a better water use efficiency and an increase in the soil organic carbon that has those beneficial effects, as I mentioned before. And to finish up, we done work with one of the most advanced cucurbit grower in the region. It's a large commercial field about 40 miles from here, where he combines multiple practices. In this case, he's using strip tillage on a stubble of wheat, and then by using the strip tillage, you incorporated the solid material, and then we go with the containerized transplants on top. And then this is using a small mini watermelon called personal watermelons, which have a higher value crops. And interesting, we observed that there was an increase in productivity, particularly in the early harvest, second harvest, and then third harvest. So the bottom line is at the end that the net profit for this grower, based on this productivity of 35 tons per hectare at the price of 330, you have a good net profit for that. In addition to that, we've seen some interesting response in terms of retention of certain nutrients in the soil, and nitrogen, phosphorus, calcium, magnesium were observed, as well as some micronutrients. So with that, I mean, the final finding on summary of this is that, we really, this humic substance particularly have improves not only earliness, but also increase organic carbon as compared to the control. And then, as we know, the soil organic carbon has a very good correlation also with water use efficiency, as well as the bacterial biomass. And we've done quite a bit of work on that. And we published this. Any one of you, if you're interested, I would be happy to share all of these publications. But it's a very interesting practice to really enhance these properties in the soil while improving quality and productivity of the crop. With that, I just wanted to thank my team, my previous teams, and several funding agencies. We're happy to maybe through the chat, because I think it's already my time is expired. But Dan, pass this to you. Thank you.
Okay, I've got one question for you. I've got a couple of the questions that came through. The one question was, are humates and humic substances the same thing?
Well, yeah, humic substance is always a combination of several, right? So you have the humic acid, fulvic acid. Okay. So
it's kind of a combination of different materials that are in that same class of substances.
Correct. The difference is, the majority of the humic acid that you see in the market, they are liquid humic acid, and the results are all over the place. I mean, this is a very unique formulation that also incorporates some novel technology on a slow release nitrogen, very, very low, very, very low. But that boost seems to have some good energy. And now we are looking, actually with the project that we have with you, we are looking at this in synergy with other potential microbials.
Yeah, okay, that's good. And then one question about water was, do you have an estimate of actual ET of the plant based on different irrigation levels?
Well, I mean, with the ET level, basically we use the weather stations, and it changes. Okay. it changes every
Yeah.
season. It changes. As you know.
Okay.